Immunology Insights, Basics and Beyond, Chapter 52, Emerging Infectious Diseases, COVID-19 and Ebola. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored influenza and other viral infections, fast-moving respiratory pathogens that test our immune system's adaptability year after year. Today, we shift from seasonal epidemics to the extraordinary events that shape human history, emerging infectious diseases. These are new or previously controlled pathogens or infections that suddenly and sometimes violently appear in human populations, often jumping from animals, and cause widespread outbreaks before immunity or medicine can catch up. Two examples define our modern era: COVID-19 and Ebola. They differ in scale and lethality, yet both illustrate a universal truth that understanding the immune system is not only about molecules and cells, but also about how quickly science can respond when nature changes the rules. So let's talk science. Our first example is SARS-CoV-2, the pandemic that changed the world. SARS-CoV-2, the coronavirus behind COVID-19, emerged in late 2019 and spread globally within months. It infected hundreds of millions and caused millions of deaths, making it the most significant pandemic since the influenza in 1918. The virus uses its spike or S protein to bind the ACE2 receptor on human epithelial cells, particularly in the respiratory tract, but also in the intestines, heart, kidneys, and vascular endothelium. Once inside, it hijacks the host's replication machinery, creating new viral particles while triggering alarm signals recognized by the innate immune system. For most infected individuals, these immune responses, led by type 1 interferons, NK cells, and T lymphocytes, effectively contain the virus, leading to mild or moderate disease. But in a subset of patients, especially older adults, the immunocompromised, or those with pre-existing conditions like obesity, diabetes, or cardiovascular disease, the immune response itself can become destructive. Instead of coordinated defense, an excessive inflammatory reaction known as a cytokine storm causes lung injury, endothelial damage, and acute respiratory distress syndrome, or ARDS. In severe cases, this inflammation spreads systemically, affecting multiple organs and increasing mortality. Children and young adults generally experienced mild disease due to lower ACE2 expression and a more flexible immune response. However, some develop multisystem inflammatory syndrome or MISC, a rare but serious hyperinflammatory condition. The pandemic also introduced a new concept, long COVID, persistent symptoms such as fatigue, shortness of breath, and cognitive dysfunction that can last for months after infection. Ongoing inflammation, immune dysregulation, and possibly viral remnants are thought to contribute to this syndrome, which continues to be studied. Despite the human toll, the pandemic accelerated medical progress at an unprecedented pace. The mRNA vaccine platforms developed by BioNTech, Pfizer, and Moderna represented a revolution in vaccinology, proving that rapid, precise, and scalable immunization is possible even against a novel virus. Our second example is the Ebola virus, a far deadlier foe. While COVID-19 became a global pandemic, Ebola virus remains mostly confined to specific regions, but is far more lethal. A member of the Filoviridae family, Ebola is a filamentous RNA virus that infects monocytes, macrophages, and dendritic cells, key players in immune communication. Once infected, these cells become dysfunctional and release large quantities of pro-inflammatory cytokines such as TNF-alpha and IL-6. Instead of coordinating an effective defense, this response disrupts vascular integrity, leading to hemorrhagic fever, characterized by internal bleeding, shock, and multi-organ failure. In fatal cases, the immune system collapses entirely. Dendritic cells fail to activate T cells, while lymphocyte apoptosis leads to immune paralysis. The result is widespread tissue damage, disseminated intravascular coagulation, and death, often within a week of symptom onset. Mortality rates range from 50 to 90%, depending on the strain and healthcare access. However, survivors mount strong and long-lasting immune responses. They produce neutralizing antibodies that can protect for years, along with memory T cells capable of rapid reactivation. These insights have been pivotal for designing Ebola vaccines, the most successful being RVSV-ZEBOV or Ervebo, a live attenuated vaccine expressing the Ebola glycoprotein. It has shown over 90% efficacy and is now used in ring vaccination campaigns, immunizing contacts of infected individuals to halt outbreaks quickly. Ebola outbreaks typically occur in Central or West Africa, linked to close contact with infected wildlife, particularly fruit bats and non-human primates. Most outbreaks remain localized and containable, but as global travel and urbanization increase, the potential for wider spread remains a concern. Continuous surveillance and community engagement are essential to prevent regional epidemics from turning into global threats. Sometimes our immune defense becomes destruction. Both SARS-CoV-2 and Ebola highlight a paradox central to immunology. The immune system that protects us can also destroy us. When viral replication is fast and overwhelming, the immune system may respond with massive cytokine release, IL-6, IL-1 beta, TNF-alpha and interferons, aimed at viral clearance but with devastating side effects. In COVID-19, this immune storm leads to leaky blood vessels, pulmonary edema, and microthrombosis. In Ebola, it causes catastrophic vascular collapse and hemorrhage. This hyperinflammation is not random. It's the immune system lock-in overdrive. Instead of resolving the infection, it becomes self-sustaining, amplifying tissue damage. Understanding this mechanism has been transformative for treatment. IL-6 receptor blockers like tocilizumab, corticosteroids, and immune-modulating antibodies are now key components in severe COVID-19 therapy, while similar anti-inflammatory principles are applied to Ebola supportive care. Just to be clear, a strong immune system is generally protective, but if it's uncontrolled or misdirected, it can become destructive, especially in diseases like Ebola and to a lesser extent severe COVID-19. Let's now look at recovery, immunity, and memory. Recovery from these diseases depends on restoring balance between viral control and immune regulation. In COVID-19, survivors generate neutralizing antibodies against the spike protein and develop long-lived plasma cells and T-cell memory, though evolving variants can partially escape these defenses. In Ebola, survivors' immune systems often produce broad polyclonal antibodies and potent cytotoxic T-cell responses targeting viral glycoproteins and nucleoproteins, a blueprint for future vaccine design. Memory formation after infection ensures faster responses upon re-exposure, yet incomplete or waning immunity can still permit reinfection or viral persistence in immune-privileged sites, such as the eyes or testes in Ebola survivors. These complexities underscore how recovery does not always mean full eradication. It means a new equilibrium between virus and host. Future directions show that we learned our lessons from the two outbreaks. From SARS-CoV-2, we learned how genomic surveillance, international cooperation, and modern vaccine technology can change the course of a pandemic. The rapid sequencing of viral genomes and open data sharing allowed scientists to track mutations in real time and adapt vaccines within months, something unthinkable just a decade ago. We also learned that public trust and equitable vaccine distribution are as vital as immunology itself. Without them, even the best science cannot stop a virus. From Ebola, we learned the importance of community engagement, early diagnostics, and ring vaccination. Public fear and misinformation can be as dangerous as the virus itself, while local healthcare infrastructure determines whether an outbreak ends or expands. Together, these lessons have reshaped the field of global immunology. Preparedness now means having platform vaccines ready for adaptation, mobile laboratories for rapid testing, and international networks for real-time immune monitoring. The goal is no longer to react, but to anticipate, to catch the next SARS or Ebola before it catches us. To conclude, emerging infectious diseases like COVID-19 and Ebola expose the fragility and brilliance of human immunity. They remind us that every new pathogen tests our ability to understand, adapt, and respond. Both viruses forced us to rethink how immunity works, how inflammation can heal or harm, how memory can protect or fade, and how science can mobilize to defend us at a planetary scale. The next challenge may come from an entirely new virus, but we now stand armed with a deeper understanding of immunity, unprecedented vaccine technology, and a global network built from experience and loss. Immunology doesn't just explain how we survive these crises, it shows how we can prevent the next one. In the next episode, we turn on one of the most consequential discoveries in modern medical history: the identification of HIV, the virus that causes AIDS. And at the center of that discovery stands a complex, brilliant, and sometimes controversial figure, Luc Montagnier, whose work changed how the world understands viruses, immunity, and global health. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time, and thank you for listening.